Hi everyone, my name is Sarah and this is a video on the Leucine Zipper, specifically of it being an example of a coiled coil. So we'll start with a little bit of a background before we jump into it. What exactly is a coiled coil? Well, a coiled coil is built by two or more alpha helices that wind around each other to form a supercoil. And there can be 2, 3, or 4 helices in the bundle. That is shown in that top image there. There's 2 of them. And these can either run in the same parallel or in the opposite anti-parallel directions. So in that bottom image, both the parallel and the anti-parallel arrangements are shown. Coiled coils contain a highly characteristic sequence motif known as a heptad repeat, in which the motif repeats itself every 7 residues along the sequence, and those are the amino acid residues, not the DNA base pairs. And the first and especially the fourth residues, known as the A and D positions, also shown there in that bottom image, are almost always hydrophobic, and the fourth residue is typically leucine, which gives it the name and the structural motif called the Leucine Zipper, which is just a type of a coiled coil. So we mentioned alpha helices. What exactly is an alpha helix? Well, the alpha helix is a common design in the secondary structure of proteins, and there are 2 major factors that help stabilize the alpha helix. The first is there's a minimization of steric interference between the side chains, meaning that they are well staggered to avoid any interference. And then they also have what's called intrachain hydrogen bonding. So there are hydrogen bonds between the NH and the CO groups of the main chain. And except for amino acids near the end of an alpha helix, all the main chain COs and NH groups are hydrogen bonded, which is shown over there on the right, hydrogen being depicted with the white circles, nitrogen as a blue, and you can kind of see the different colors better over there in the way that they are bonded in that way. And this results in hydrophobic interaction, meaning that there's an observed tendency of nonpolar substances to aggregate in an aqueous solution and exclude water molecules. So these are some of the factors that make these alpha helices in the coiled coil both very stable and very strong. So this picture shown here was given to us in this prompt. The Leucine Zipper shown right is an example of a coiled coil. Again, a typical Leucine Zipper consists of 2 alpha helices with the leucine residues in about 7 residue intervals, and shown right in that red box is one of those intervals. So our first prompt was what possible residue could replace leucine and also maintain that hydrophobic core? So I am choosing isoleucine as one residue that could replace leucine and maintain the hydrophobic core. And there's a couple of reasons for that. So there's 2 pictures shown on the left. That top one shows that they have a similar number of those C atoms in their side chains, and that's depicted in the red. And so because of that, they're going to have similar bonding. And then I listed a few of their common properties below. They are both aliphatic, they are both hydrophobic, and they both have that neutral charge. And so I think that isoleucine would be able to adequately replace leucine. And then our second prompt was, well, what's a possible residue that would then kind of do the opposite and destabilize that core? And I believe that lysine would be one that would actually destabilize the hydrophobic core. And I believe that because of the polar interaction in the core itself of lysine. And so in kind of layman's terms, lysine is becoming charged. And I showed that though here in those 2 images, leucine being on top. And you can see that some of the properties include that it's hydrophobic and neutral, whereas all of a sudden you have lysine shown there at the bottom, which is hydrophilic and a positive charge. That's going to disrupt that core that leucine had going on, and that's going to kind of alter the structure and the function in that way. And so lastly, our prompt was, what amino acid pair could interact in a similar manner to lysine and glutamate, specifically their side chains. And so I believe that aspartate and histidine, that combination would be a good pair that could interact in the same manner to lysine and glutamate. And I think that because they share some of the same properties of together having one basic side chain and one acidic side chain, and they are all polar and hydrophilic, so they're able to be swapped kind of in and out in that way. This is depicted better over there on the right, and I color-coded them to make it a little bit more clear. So you have the pair that we're comparing them to, lysine and glutamate at top, and you can see their structures as well as their properties that I highlighted. Lysine being that charge, that positive charge, and glutamate being that negative charge. So at that bottom then, histidine would be able to be the same positive charge in a sense as lysine, and aspartate I'm thinking could be that same negative charge as glutamate. And then as you can also see, they all have the same properties of being both polar and hydrophilic. So in that way, I believe that aspartate and histidine would interact in the same manner to lysine and glutamate. Thank you for watching. I hope you learned a little bit more and also a few new ideas related to the secondary structure and nature of proteins and the leucine zipper as being an example of a coiled coil. I look forward to watching your videos and hearing your thoughts on this presentation. Thanks!